Thank you for joining us on today's Coffee Break webinar. Today's topic is Restoring Fragile X Neuron Function with Gene Editing. Fragile X syndrome is the most common genetic form of intellectual disability with an incidence of 1 in 4,000. Patients with Fragile X syndrome display a broad spectrum of autistic phenotypes such as intellectual, social, and reasoning deficits. These characteristics are attributed to the loss of the Fragile X mental retardation protein, or known as FMRP, encoded by the FMR1 gene during brain development. FMRP is an RNA-binding protein which is essential for the maintenance of normal synaptic function. Currently, there is no cure or treatment for Fragile X syndrome, due in part to the enormous complexity of the Fragile X syndrome neuronal circuitry. In this webinar, Dr. John Graef, principal scientist at Fulcrum Therapeutics, demonstrates how using patient-derived cells and CRISPR gene editing can create and reverse the Fragile X syndrome phenotype observed in a dish.
Thank you, Melissa, for that introduction. What I would like to talk about for this webinar is how at Fulcrum we are using patient iPSC-derived neurons and Axion's multi-electrode array platform to elucidate a functional phenotype for Fragile X neurons, and how we went about correcting that phenotype by restoring levels of FMRP, the deficient protein underlying this neurodevelopmental disorder. First, I'd like to tell you about Fulcrum Therapeutics. We are a drug discovery company located in Cambridge, Massachusetts, and our focus is on treating the genetic root cause of diseases. Our fundamental premise is that by working on monogenetic diseases caused by disrupted gene regulation, we can focus our drug development strategies on normalizing the aberrant gene expression. One of the diseases we are working on is Fragile X. Fragile X syndrome is a neurodevelopmental disorder with an incidence of 1 in 4,000. It is the major genetic cause of intellectual disability and mental retardation. Patients with Fragile X also display a variety of autistic spectrum phenotypes, including anxiety, avoidance, and verbal disability. Almost 4 decades of Fragile X research has identified the X-linked gene FMR1 as the disease-causing gene. The majority of Fragile X patients have an expanded CGG repeat at the 5' untranslated region of the FMR1 gene, which leads to the epigenetic silencing of FMR1 and therefore no expression of the protein FMRP. FMRP is an important RNA-binding protein localized in the synapses and therefore plays a critical role in neuronal communication. To date, the most relevant preclinical trial for Fragile X is based upon the mGluR5 theory. This theory argues that FMRP plays a negative role in activity-dependent local protein synthesis associated with the metabotropic glutamate receptor mGluR5. However, several clinical trials targeting mGluR5 have failed. These could have failed for a number of reasons, but the mechanisms are all acting downstream of FMRP, acting at proteins whose expression is themselves controlled by FMRP. Fulcrum strives to correct the root cause of Fragile X by reactivating FMR1 and FMRP to physiologically meaningful levels. We feel that by restoring enough FMRP to neurons, we can correct the functional deficits. However, before we can do this, we need to identify a functional phenotype in Fragile X patient-derived neurons and then determine the FMRP levels needed to correct this phenotype. In order to identify a functional Fragile X phenotype, we have used Axion's Maestro MEA platform to measure differences in spontaneous activity between isogenic pairs of iPSC-derived neurons where FMRP expression is the dependent variable. We are utilizing the NGN2 method previously described by Marius Vernig and Thomas Sudoff to differentiate iPSCs to neurons through overexpression of the transcription factor neurogenin-2. An example of spontaneous spiking activity from 16 different electrodes in one well can be seen on the left-hand side of the slide. On the right is an example of spontaneous activity shown on a 48-well MEA plate as depicted by a real-time heat map. We use 3 different isogenic cell lines to assess the FMRP-dependent effects on functional activity on the MEA. These 3 isogenic lines are shown here. The first pair of cell lines, denoted as SWFSX and SWC12, were generated by Steve Warren through CRISPR-edited removal of the CGG repeats in a Fragile X patient line. The immunofluorescent images for these neurons are shown on the top left, where the nuclei are shown in blue, the neuron-specific microtubule component beta-tubulin is shown in green, and FMRP is shown in red. As can be seen from the images, both cells form viable neuronal networks, with the Fragile X neurons having no FMRP expression, and the CRISPR-corrected neurons showing high levels of FMRP located in the cell body. The second line used was a wild-type line purchased from Thermo Fisher denoted as EPI49. A FMRP knockout cell line was created from this wild-type line by Dr. Hao Wu, a senior scientist in the biology group here at Fulcrum, by inserting a premature stop codon in FMR1 in which FMR1 mRNA levels are still measurable, but no FMRP is detected. Images for the wild-type neurons, which show detectable FMRP levels, and for the FMRP knockout neurons, which don't, are shown to the left. The final isogenic pair used was also generated from a Fragile X patient line, denoted as 135.3. This line was also generated by Dr. Wu through CRISPR editing of the CGG repeats in order to produce a clone with a truncated CGG repeat region. Images of this isogenic pair also show detectable levels of FMRP in the CRISPR-corrected isogenic control neurons and a lack of FMRP expression in the Fragile X patient-derived neurons. On this slide is a figure from our recent Cell publication demonstrating the method for deleting CGG repeats in the FMR1 locus with CRISPR-Cas9 in order to reactivate FMR1 expression in the C12 line. Panel B shows FMR1 mRNA levels measured by qPCR in a control line, the Fragile X line, and the isogenic C12 generated by CRISPR-Cas9 editing. Panel C shows representative immunofluorescent images of these three lines with DAPI stains shown in blue, beta-tubulin stains shown in green, and FMRP shown in red. Panel F demonstrates that re-expression of the FMRP in the isogenic control neurons with truncated CGG repeats show reduced excitability as measured by spikes per minute over a 2-week period on the Axion Maestro MEA system when compared to the Fragile X neurons. In follow-up experiments, we used the 3 previously described isogenic lines to confirm this increase in spontaneous activity on the MEA. We were able to demonstrate hyperactivity in neurons lacking FMRP that manifested after 3 weeks in culture for all 3 isogenic pairs tested. These experiments established a functional phenotype for Fragile X iPSC-derived neurons where reduced FMRP levels leads to neuronal hyperexcitability. In these graphs, the neurons lacking FMRP are shown in blue, and the neurons expressing FMRP are shown in gray. The x-axis denotes the time in culture, and the y-axis shows the average weighted firing rate per well, which was calculated as the product of the spontaneous spikes per minute and the number of active electrodes in the well. Once we had established a functional phenotype and knew that restoration of FMRP to near full wild-type levels was enough to normalize this increased spontaneous activity, we wanted to determine if partial restoration of FMRP was sufficient, and if so, what partial FMRP levels are needed to correct the functional phenotype. We did this 2 ways. First, we asked if full restoration of FMRP levels in only a subset of cells was enough. We used FMR1 mRNA transfection experiments, along with experiments where we titrated in FMRP-expressing cells in varying percentages into Fragile X cultures. These experiments essentially created FMRP mosaicism in a dish. Next, we asked if partial restoration of FMRP in a majority of Fragile X neurons was enough. We did this by titrating in varying MOIs of a virus containing dCas9 TET1 and CGG guide RNA constructs to achieve varying levels of FMR1 demethylation and reactivation. This slide shows representative immunofluorescent images demonstrating FMRP mosaicism in a dish. The top 3 images are from Fragile X neurons 24 hours post-transfection with FMR1 mRNA. As can be seen from the top middle image, only a subset of Fragile X neurons took up the FMR1 mRNA and are expressing FMRP at near control levels. Additionally, the bottom 3 images show examples of Fragile X and control neuronal cultures where FMRP-expressing neurons were mixed with Fragile X neurons. The bottom middle image shows a culture where 20% of the neurons were expressing FMRP. This slide summarizes the data from our FMRP mosaicism experiments. Starting in the top left-hand corner part of the slide, this graph shows the average number of FMRP-positive neurons 24 hours post-FMR1 mRNA transfection in Fragile X neurons as measured by immunofluorescence. The data are normalized to the isogenic control shown in dark gray, which was set at 100%. This shows that when neurons were transfected with 4 to 12 nanograms of FMR1 mRNA, depicted as the green, blue, and orange bars, that this leads to around 5 to 10% FMRP-positive neurons. When neurons were transfected with 16 to 24 nanogram FMR1 mRNA, shown by the brown and gold bars respectively, this resulted in around 20 to 25% FMRP-positive neurons. The corresponding MEA data is shown below in the bottom left-hand corner. This graph shows increased spontaneous activity for Fragile X neurons in light gray as compared to the isogenic control neurons shown in dark gray. The spontaneous activity of Fragile X neuronal cultures transfected with FMR1 mRNA levels that lead to 5 to 10% FMRP positive neurons shows a trend towards reduced activity. However, Fragile X neuronal cultures transfected with FMR1 mRNA amounts that lead to 20 to 25% levels of FMRP positive neurons show full normalization of hyperactivity. The data from experiments in which FMRP expressing neurons were mixed with Fragile X neurons is summarized in the upper right portion of the slide. Here, the increased spontaneous activity in Fragile X neurons in the gray bar over control neurons in the black bar shows a partial normalization when less than 10% FMRP expressing cells are mixed into the culture. Only when 20% or greater FMRP expressing neurons are added is the hyperexcitable Fragile X phenotype fully rescued. Therefore, transient transfections of FMR1 mRNA and titration of control neurons into Fragile X cultures suggest that 20% of neurons need to be expressing high levels of FMRP to normalize hyperactivity. This slide shows another figure from our recent Cell publication demonstrating the method used to demethylate the CGG repeat region of the FMR1 gene. An inactivated Cas9 enzyme was tethered to a TET1 enzyme and targeted to the hypermethylated CGG FMR1 repeat region with CGG guide RNA. This led to demethylation of the FMR1 CGG repeat region, reactivation of FMR1 levels, and normalization of hyperactivity as measured on the MEA. This was done in iPSCs followed by FACS sorting in order to capture successfully transduced cells, then differentiated into neurons to assess FMR1 expression, shown in the lower left, and spontaneous activity, shown in the lower right. The MEA data shows that over time, the increased spontaneous activity seen in the Fragile X neurons, shown as the red line, as compared to the control neurons depicted as the blue line, can be rescued following near full restoration of FMR1 levels. In follow-up experiments, we transduced post-differentiated Fragile X neurons with the same dCas9 TET1 construct to determine if low levels of FMRP expression in a large portion of neurons could also normalize the increased spontaneous activity. The immunofluorescent images in the upper right-hand corner show that after transduction with the dCas9 TET1 construct, low levels of FMRP could be detected in the majority of Fragile X neurons. This is quantified in the lower left as around 5 to 7% FMRP levels. The MEA data is shown in the upper right-hand corner of the slide. This data demonstrates that Fragile X neuronal cultures that are expressing 5 to 7% FMRP levels begin to reduce this increased spontaneous activity. Therefore, demethylation of the CGG repeat region through transduction of dCas9 TET1 suggests that at least 5% FMRP levels in a large proportion of neurons is needed to normalize the Fragile X hyperexcitability phenotype. In summary, reduced FMRP levels lead to neuronal hyperexcitability as measured by the Axion MEA system. Close to full restoration of FMRP levels in around 20% of Fragile X neurons is sufficient to normalize this hyperexcitability. And then greater than 5% FMRP restoration in a majority of Fragile X neurons is sufficient to normalize the increased spontaneous activity seen. In closing, I would like to acknowledge everyone who was instrumental in generating this data. Thank you for your time, and I hope you enjoyed this webinar.
And that is the conclusion for today's Coffee Break webinar. If you have any questions you would like to ask regarding the research presented, or if you are interested in presenting your own research with microelectrode array technology, please forward them to coffeebreak@axionbio.com. For questions submitted for Dr. Graef, he will be in touch with you shortly. Thank you for joining in on today's Coffee Break webinar, and we look forward to seeing you again.